Hey everybody, hope you are fine. Today I'm with the Chris, and Chris is going to talk about his PhD project. And before that, can you explain your background in a few sentences?
Yes, so hello, I'm Krzysztof Jurdziński, and I'm a biochemist by trade. I've done it in Krakow, my undergrad in my home country of Poland. Then I've done a master program together with Cenk here in Stockholm, molecular techniques in life science. But now I ended up in environmental science. So I study ecology and evolution of bacteria in different water bodies. So in lakes and in the Baltic Sea, which is one of the seas, but it has less salt than other seas, and we do also look at a lot of other seas. So we do study microorganisms. Those are organisms that you cannot see with a naked eye, but only with microscope. We do not use that microscopes that much. However, we mostly look at DNA. So we look at DNA we take a sample, a bit of water, we get all the DNA from there, and with a bit of computation laboratory and computational magic, we get information about all the different organisms that lived in the environment, most of which you cannot cultivate in a laboratory, you cannot grow in laboratory. And also how many of which organisms were there. So what I do, I compare organisms from different environments, mostly related to salt concentrations, so freshwater lakes, seas, and the different weird bodies like the Baltic Sea that I mentioned that has intermediate salt concentrations. I'm also involved in a biodiversity monitoring program where we look at how many different microorganisms, microbes are at different times of the year and in different places along Swedish coastlines, and we want to make a framework to be able to be monitored in long term across years, see impact of different mostly human induced changes on all the microorganisms on a wider scale. And one more important question that we are interested in is how bacteria disperse. So we know they often are the same kinds of bacteria all around the world, but we don't know how that happens because they are small and they don't travel on their own. What do? Maybe they do fly with the air. We actually want to look at that.
Now, how is your one typical day as a scientist?
So I do most of my job actually on the computer with data. So I get to my office, I get myself a tea, I do a bit of coding. So I program mostly in a language called R. I write. Then I look at my results, mostly graphs. I think about them. I write. Maybe I read something, or maybe I talk to my supervisor. Then my view on my results changes 180 degrees. So I come back. I do a bit more coding, and I look at the results again. I go home thinking about what I'm gonna write about them today.
And what is one of the most interesting results you got so far from your project?
Okay. So one of the most interesting results I got, I looked at some changes in bacteria which switch to different environments when it comes to salt concentrations. And we estimated how long did it take for them. And bacteria are small, they divide very rapidly. And then we see that there are changes so big in how they function that it takes probably millions of years, probably tens of millions of years if our estimations are correct. But even if we are not right an order of magnitude, it's a lot of time. So that was very interesting. Like,
Thanks for sharing your PhD project, and wishing you good luck in the next years.
Thank you, Cenk. And thank you for listening.
Take care.